Are we good? Let's start. Okay. Good afternoon, everyone. Thank you for joining us for our final seminar of this semester with Dr. Paul Scott. He is joined here today with his family. He has asked me to keep the introduction brief, not to take away from his presentation. So he spent 29 years working at USDA ARS as a research geneticist, and I'll let him give his presentation from there.
That was great. Are we still on? Are we doing okay? Yeah, so it's just a great honor to be here to talk to you today. This, been my home for the last 29 years, and it's so nice to be invited to give a seminar here. It turns out when they ask you to give a seminar and at the end of your career and you're retired, they say, you can say anything you want. And what an opportunity that is, right? So I'm gonna have to be a little careful. But so I thought a lot about what I wanted to say today, and really the main thing that I want to say is thank you to everybody who made my career such a wonderful experience. And so the title of this talk kind of refers to that. If I've seen further, it is by standing on the shoulders of giants. That's something Isaac Newton said a long time ago, and I really like that quote because it really is a great analogy for how we do things in science, right? You're always building on the work of others who went before you. And so in this talk today, I want to talk about, I'm just going to kind of give an overview of my career and try to acknowledge some of the people that had a real big impact on me. And I also want to leave you with some lessons that I learned that I think are important and I hope will help you in your careers as well. So let's see, I've never interacted with this device before, so okay, there we go. Yeah, so the first lesson that I want to talk about is the importance of mentors and role models. And when I say mentors and role models, I acknowledge that there are formal mentoring programs and those are good, but I would say more important to me have been the informal relationships that aren't really designated as a mentor-mentee relationship, but right, it's my interactions with colleagues and from all aspects of work that I've taken away important information from that I've been able to incorporate into my work style that helped me. So the lesson here is pay attention to everybody around you, not just the lead scientist in your area, and try to learn from everybody you interact with. So I want to tell a story about my first role model, and so this is how I got interested in crop science as a teenager. And I was 16 years old in 1980, and I went to Ames High School, and I was a wrestler at Ames High. And I don't know if you can figure out who I am. This is me right here. That's funny. So I wanted to get better, and Iowa State University at the time was consistently in the top 5 nationally ranked wrestling schools in the country, and they had a wrestling camp every summer. And I went to this wrestling camp, and it was a great experience. They brought in Olympic wrestlers and all former ISU people and taught us a lot of things about wrestling, and it was a really good experience. And in the end, sometimes it's hard for me to get through this because it's a little kind of emotionally charged for me, so I apologize if I'm getting a little choked up here, but in the end of this camp, they got everybody together, 100 wrestlers from across the nation, and Dr. Harold Nichols, who was the ISU wrestling coach at the time, came out, and this guy, he was maybe 70 years old at the time, the most successful wrestling coach of up to date, and he addressed the camp. And he didn't talk to us about wrestling. What Harold Nichols talked to us about was plant breeding. Because Harold Nichols went to the small town of Cresco, Iowa. It's a little town in northern Iowa, and one of his teammates was a guy named Norman Borlaug. So this is Norman Borlaug back in 1932. And of course, you all know Norman Borlaug, that he was the father of the Green Revolution. He developed high-yielding wheat and rice varieties that were grown in Southeast Asia at a time when there was projected to be a great shortage of grain. And he's credited with saving 1 billion lives by developing these high-yielding wheat and rice varieties. And I thought, you know, as a young wrestler, the idea that an Iowa farm kid who was a wrestler just like me could save 1 billion lives, that was so motivating to me. It was just really impactful. And so I decided, that's when I decided I wanted to get into crop improvement, to do something along those lines. So that's my story of how I decided to become a plant breeder. And I went to Iowa State for my bachelor's degree. I studied biochemistry, and we didn't have a genetics program then. It developed while I was a student, but biochemistry was as close as I could get to genetics. And I did some undergraduate research with Jack Horowitz. We studied the structure of tRNA, and we used NMR, and it was a really good project for an undergraduate student. I worked with David Morris, who was a molecular biologist who worked on corn. In those days, Don Robertson was one of the most famous corn geneticists around. He studied Robertson's mutator, the transposable element that maybe some of you know. And he worked in the ISU genetics lab, and they hired David Morris to do molecular biology to complement his genetics work. And I worked in David's lab, and that was a great experience. And then my first professional job was actually an internship that I took at Pioneer Hybrid. And I worked for, my supervisor was David Grant, who is here in our department, right? So that's a cool thing that David is still here. And what I did was I spent a summer in his lab, and we purified DNA from corn plants. I did that for a whole summer. And we kind of thought that we had probably, our little group, purified DNA from more corn plants than anybody had ever done previously. So, you know, that's kind of a, you know, personal best kind of thing. We did 500 DNA extractions in one summer. So I'm sure some of you have done 500 DNA extractions in one afternoon, right? So things change a lot. Okay, so that was my undergraduate research. I went to graduate school, and I had 3 great advisors in graduate school. Niels Nielsen at Purdue University was a USDA scientist like me, and the important thing that he taught me was about scientific standards. So, you know, you can publish almost anything if you work at it, right? You can get publications out. But Niels always insisted that we would aim high in our research, and we were always shooting for the top-notch publications and the best in the best journals. And just the idea to a young graduate student that there's good publications and bad publications, that was an important idea to me. And I hope you guys have figured that out and are working to get those good publications. I went to Denmark next, and Bia was a great leader in his group. I really enjoyed his leadership style. He had a large lab of about 30 people. But the thing that really struck with me about his career is his persistence. So he started as a graduate student studying cyanogenic glucosides in plants. These are toxic compounds that make plants toxic, and obviously important agronomically. He worked his whole career on cyanogenic glucosides, which is a fairly focused area. He published more than 400 papers on this subject. And of course, he's the leading expert in that. And the reason he was able to accomplish so much is because he kept focused on the same problem for his whole career. And that's remarkable to me. And then my second postdoc at Lincoln, Nebraska was with John Goldbeck. And that was a great project because we were doing biophysics, and we would develop these instruments that had lasers firing through spectrometers and stuff, and it was really an interesting process. But we generated tons of data, and we had to come up with ways to manage these large volumes of data. And the ideas that I developed in that time about data management and data integrity became really important later on when we started getting DNA sequence data and we started doing genomics research. We had the same problem, right? Tons of data, and how do you maintain the integrity of that data and keep it organized? So those were things that I took away from my advisors in graduate school and postdocs. They all had different leadership styles, and when I started in the USDA, I kind of picked and chose from my advisors' leadership styles and kind of tried to synthesize my own leadership style. And that worked okay, and I think people should do that. It's an easy way to develop a good leadership style. But then I figured out that with training and reading books on the subject, you can actually learn the process of leadership and personnel management. And this is actually, this was a revelation to me, that leadership is a learned skill. And I encourage you to work at learning leadership and personnel management if that's something you're interested in, because you don't have to be born a natural leader to be a leader, okay? So that was I think a useful lesson that I learned along the way. And then the other lesson I want to give you at this time is I like the idea of focusing on the problem. You can have more of an impact if you stay problem focused rather than focusing on the methods that you're using. A lot of people kind of let the methods drive their research interest, and I think you can be more impactful if you focus on the problem rather than the methods. And just to illustrate that, I have these pictures of me back in graduate school doing a DNA sequencing experiment. So I think I must have sequenced 1,000 bases of DNA in graduate school, and that to me seemed like a pretty big effort. But we had to use radioactivity, so this little thing here, maybe that doesn't look familiar to many of you, that's a rad badge, so you can keep track of how much radiation you're exposed to. And we would use these terrible sequencing gels. They were these homemade contraptions that, you know, they had two chambers that held liquid, and then you would put a gel in there and connect these electrodes to it and put 3,000 volts of electricity across those chambers, and the DNA would fly through this gel, and if you were lucky and didn't get electrocuted, you could sequence 100 bases in a run or something like that. And so this is what DNA sequencing looked like in those days, and you know what it's like now. I don't even know what happens. You mail a test tube of a piece of a plant off and you get a genome sequence back pretty soon. It's really different. Yeah, so methods change, but the problem stays the same. And the problem that I decided to work on when I took this job was amino acid balance. And I still have the slides that I used when I gave my interview presentation here in Ames for this job. And this is one thing I talked about, was the problem of amino acid balance. And then about 2 months ago, I gave a guest lecture in Emily Heaton's plant agronomy class at University of Illinois Urbana-Champaign, and I used this slide, which is almost the same as the slide I used in my presentation 29 years ago. So I guess I did a pretty good job of staying focused on this problem of amino acid balance, and I want to talk to you about it, not because it's the most impactful thing that I did, but I think there's some interesting lessons in it. So the idea with amino acid balance is that animals require 20 amino acids in order to do growth and metabolism, right? 10 of them they can interconvert with biochemical pathways, but 10 of them are essential and they have to be in their diets. And this graph in green shows the level of all the amino acids relative to the needs of animals. So if in a perfect nutrition, in a perfect protein source, all the amino acids would be lined up at 1, but we have some deficient amino acids in corn, like lysine. Boy, it's hard to give a talk when you're shaking, and using the touchpad tends to go off. And I don't suppose there's a mouse around, right? It's oh yeah, that's what I can't use those to point though. Okay. Anyway, amino acid balance. We don't need to increase the amount of protein in our crops. What we need to do is improve the amino acid balance. So just a few amino acids like lysine and tryptophan, if we could improve the level of those, we would increase the value of all the protein in the plant. So that's kind of the idea that I've been working on for much of my career. And I took 3 different approaches to solving this same problem. And they're mutation breeding, recurrent selection breeding, and then biotechnology. And I just want to talk for a few minutes about each of those different approaches, starting with biotechnology, because I was really gung-ho about biotechnology when I started. And I was really lucky that ISU had just created the nation's first plant transformation facility under the direction of Con Wang. And I had great cooperators like Larry Johnson, Chuck Glatz, and Mike Lee. And we together put a lot of different genes into corn plants and learned a lot by doing so. So one of the first things we put in was a milk protein called alpha-lactalbumin that had a really good amino acid balance, and it was naturally present in milk. We took the sow's milk version of it and made a corn gene and put it in, and it worked great. This, thank you. Oh, this is nice. Yes, it works. Yeah, so this worked great. We increased the level of lysine in corn plants by something like 46%. It worked really well. We did some other fun stuff too, like we put in a wheat gene called 1DX5 into corn. And that was cool because, you know, wheat proteins are important for gluten intolerance, which is a huge problem today, right? And here was a system where we could isolate one individual component of wheat gluten and study it in a background that's non-gluten intolerance inducing. So in wheat, there's like 100 genes that contribute to gluten intolerance, but in this corn, there was just one. So what a great system this was to study gluten intolerance. And then the other thing that we thought we could do is wheat proteins cause dough to be extensible and elastic. Like if you think about the difference between wheat bread and corn tacos, right? When we put this gene from wheat into corn, we could tell a difference in the dough when we would mix it with water. It would make a little dough ball. And we thought we could make corn tortillas that would be foldable without cracking, less tendency to crack, right? So we thought this was a cool idea. We had a lot of ideas like this. We talked about a product we called tortukis that we would make out of this corn wheat flour, and maybe that's what I'll bring to the holiday party is some tortukis. Yeah, we've never made them. So anyway, we were doing nice work with biotechnology, and suddenly the world, almost overnight, changed. In the year 2000, Starlink was not a satellite network. Starlink was the name of a BT corn event, and this BT corn event was regulated such that it could be used in animal products but not in human food products. So that was a big problem, and it wasn't that this event was toxic in any way. Nobody ever had as much as a headache from eating Starlink corn, but it didn't have the regulatory approval, and when it started showing up in taco shells and other places, the world changed and freaked out, and transgenes became something that people were very worried about. Everybody was worried, is there going to be a transgene in my cereal? You know, it was something that people thought about theoretically, but when it started happening, it really changed the landscape. So USDA evaluated the work that I was doing and decided it was too risky, and they asked me to stop doing this work because of the public, you know, the dangers of the public perception, right? Which was a really unusual thing for the USDA to do. So I guess this led me to a lesson, another life lesson. Pay attention to current events and respond appropriately, okay? It's important for your research to be relevant in that way. So I changed the focus of my research a little bit. We kept working with transgenes, but we started working with marker genes. So we put in GFP, this protein from jellyfish that glows. And we used that to try to understand a little bit more about gene expression, because people were saying all kinds of crazy things about transgenes. They were saying, we have no control over the expression of these genes. We put them in and we don't know what's going to happen. And, you know, I didn't think that was true, and I don't know, I hope you don't think that's true either. But there was very little data to show that scientists actually did understand how transgenes might function in a system. And so I tried to do some real simple experiments just to show that if we make a transgene that expresses GFP and it's based on a corn seed protein like zein, then the transgene is going to behave a lot like the zein gene does, right? And so we made a transgene that was regulated by opaque-2, just like the zein genes do. And we made transgenes that we could actually select for the transgene expression. So corn kernels that glowed more intensely, and those kernels had a higher level of genes that had the same promoter. So you could select for a native gene by making a transgene that looks like it, select for the transgene, and the native gene could go up too. So that was pretty cool. Okay, enough of biotechnology. Moving on to recurrent selection breeding for improving the amino acid balance. And at the time I started this project, Kendall Lamke was the lead scientist on my ARS CRISP project. And Kendall was always telling me, if you can measure it, I can select for it. And so one Christmas, when everybody was gone, I went into the lab and I figured out how to measure amino acids in a high throughput way that a plant breeder could use. And I based it on a microbial method, similar to this one that actually Alan Wright was the scientist who had my position before I took it. So he had a good idea about amino acid balance. And so the way these microbial assays work is you start with a bacterial strain that has a mutation in its biosynthetic pathway for an amino acid like methionine. And that bacterial strain requires that amino acid to grow. And it turns out that you can provide these amino acids in a corn extract. And the growth of that bacterial culture will be proportional to the amount of the amino acid in that culture. You can design this experiment so the growth is limited by the amount of methionine in the assay. So I'm showing this in test tubes, but we did this in 96 well plates, and this was a really high throughput method, and we did do thousands of these assays in a few months, and they cost really just the highest cost was the labor to grind and weigh the corn. So almost no cost. And the competing technology is HPLC, which costs $100 a sample and takes hours to run. So we turned the crank on these assays, and we did selection for methionine levels in a couple of different corn populations. So what I did here was we did divergent selection. So we selected for high methionine corn and low methionine corn. And then I pooled the high and low selection experiments for each population and analyzed them all together. And the light blue bars are the low methionine population individuals, and the dark blue bars are the high methionine population individuals. And you can see that the two populations, which started out at the mean, have now diverged almost one standard deviation, high and low, from the mean. And they did that in two different selection experiments. We did it in BS11 and BS31, two Iowa State populations that we liked. So this worked great. And since then, we've derived doubled haploids from these high methionine populations. And those inbreds are some of the highest methionine inbreds that have been reported anywhere. So those are available for someone who needs a high methionine source of corn. Okay, that was recurrent selection breeding. I want to talk just a few minutes about mutation breeding. Same problem. How do you improve the amino acid balance with mutation breeding? Well, there's a mutation called opaque-2 that confers high lysine to corn, and it's been known since the 60s. And the problem is that mutant kernels are soft and they break easily. Insects eat them, and so they're just not agronomically adapted. So for 30 years, researchers at CIMMYT worked on selection of hard versions of the opaque 2 kernels that had high methionine trait we were interested in. And Surinder Vassal and Evangelina Villegas got the World Food Prize for developing what they called QPM, quality protein maize, that had agronomically adapted kernels with high nutritional value. And CIMMYT has done a great job of releasing QPM to a lot of places in the world, except temperate areas. And so I had the good fortune to work with Arnold Hollauer. After he retired 2 or 3 times from Iowa State University, he decided he wanted to work on quality protein maize and develop a temperate version of quality protein maize. And so I worked with him on this and helped measure the amino acids and evaluate the kernel hardness. You can see the kernels segregating here, sorry. For kernel hardness, you can see the light comes through some of them and it doesn't from others. That's the hardness trait that we were selecting for in quality protein maize. And we released these, and quite a few people have requested them. I think on 3 or 4 continents we've had requests for this germplasm. And it was also useful in some experiments, like my cooperators, Clark Ford and Erica Fuchs, used them to identify the modifier genes that cause this kernel hardness phenotype. So that was useful material. Okay, so looking back, I told you how we solved this problem of amino acid balance 3 different ways, right? So surely the United States corn no longer has problems with amino acid balance, right? That would be wrong. I'm sorry to say that in spite of our efforts to improve amino acid balance, corn breeding companies have been selecting for yield and moisture. And over time, this is a study that Jody Edwards and I did with some cooperators from Pioneer. We looked at Pioneer hybrids that were released in different years from 1920 to 2000, and over that time, what happened was starch increased, protein decreased, and oil decreased. And we looked at amino acids, and they had just a very, probably not significant decrease over this time. And it turns out that this makes corn great for biofuel production, right? You want a lot of starch, a little bit of protein, and yeah, that's almost perfect for biofuel production, which, interestingly, is exactly what happened to the corn market during that time. So corn production has been going up, and look at what happens to biofuel production. It went from, at the start of my career, it was just a few percentage of the corn market, and now it's up. Almost half of the corn goes into biofuels today. So the large seed companies were responding to this rapidly growing demand for biofuels. Of course they were. That's what you'd expect them to do, right? And so for the first 15 years of my research, I thought my stakeholders were, these large seed companies. But in fact, that was not right. And I had to think about, really who is my research important to? Who are my stakeholders? So this is just kind of a summary of what I just said. And I don't want to leave on a negative note about amino acid balance, because QPM has been adopted in a few places where it's having a big impact. And especially the bright spot I see is there's a company called Semilla Nueva that was started by my friend Kurt Bowen in Guatemala, and they're doing a great job of releasing QPM and fortifying QPM, even with genetically fortifying it with zinc as well, and releasing that corn in Guatemala, and he's starting to look at Africa now as well. So this is really the bright spot, I think, in improving amino acid balance. But the lesson for me out of this years of work is you really have to know your customers. So I thought a lot about who are my customers, who cares about nutritional quality in corn, and the answer I came up with was one group that cares about it is organic farmers. And I was lucky back in around 2010 to work with a USDA colleague named Linda Pollock. Here she is. Linda wrote a grant that was funded by the NIFA organic research and education institution. And it was for developing corn for organic farmers. And so I worked with different groups of people in this grant over time, and we had several iterations. I think we had 4 different grants from this program, and here's the current group. And this grant just ended, but this group is planning to put in another proposal, I think, to the same program. So I really enjoyed this work. It was impactful work, and it was nice because my customers really appreciated the products we were working on. It also led to kind of a branch project for me, because I'd been focused so much on nutritional quality. And I talked to these organic farmers, and they said, yeah, we like the nutritional quality, but we have this other problem too. And it's that transgenes keep blowing into our fields from adjacent neighboring GMO cornfields. And I guess maybe there's a little bit of irony in that they wanted me to develop a method to keep the GMOs out of their organic cornfields. And that's how we started getting into the idea of working with cross-incompatibility. So there are genetic systems that confer cross-incompatibility. There's one in popcorn where you can pollinate dent corn from a popcorn plant, and it works fine. It sets seeds like normal. And popcorn can self-pollinate, of course, but dent corn cannot pollinate popcorn. So this is just what you want to control the movement of genes around, where you have it's basically a unilateral cross-incompatibility system. It goes one way. And so we decided to try to use a system like this, and other companies also worked in this area. We weren't the only ones in here, but the idea was we could backcross the GA1 system from popcorn into standard dent corn, and it would prevent the pollination of organic corn by GMO pollen. And we had a lot of challenges because the molecular characterization of the genes had not been done, right? So we had to do, it was a difficult phenotype to measure, and we didn't have molecular markers or anything. So one of our top priorities was to figure out what this gene was. And it has a really interesting history because people have studied this gene for more than 100 years, right? The first report is by Karl Korens, who was a German botanist in 1901. And what strikes me is that Korens is one of the people who was credited with rediscovering Mendel's laws around 1900. And I'm trying to imagine how Korens was looking at this data from GA1 and also looking at Mendel's laws. And GA1 totally messes up Mendel's laws, right? It doesn't work at all. And somehow he was able to rediscover the value in Mendel's laws, even though he was studying GA1 at the time. I thought that was pretty weird. Okay, so there's a lot of really high-powered maize geneticists who worked on this gene in the 100 years before we started working on it. And they pretty clearly had genetic evidence to say there are 2 functions at this one locus, and those functions are absent in almost all of the corn, any of the corn that doesn't have the GA1 locus. So everything is not popcorn. It doesn't have this allele, it's just a null allele. And a lot of people were doing transcript profiling to try to track this gene down, and they were failing, and we think it's because they were using B73 as their genome, their reference genome, and B73 doesn't even have the locus in it, right? And we knew that genetically. So we took a different approach, and in 2017 we published characterization of this pectin methylesterase that's responsible for GA1 activity, and we identified it by doing de novo assembly of the transcripts rather than mapping them to B73, which we knew didn't contain the gene we were interested in. And using genomic de novo assembly, Dr. Adrian Moran-Lauder in the audience was able to clearly, it really stuck out like a sore thumb, this pectin methylesterase. And this was really, I would say, the highlight of my career when we made this discovery. I don't know, probably maybe the highlight of your career as well, maybe. Yeah. Anyway, it was a really cool thing because Adrian gave a talk at the Maze Genetics Conference, and a lot of people who'd been working on this gene, some of them had been working on it for 50 years, and we got to report the molecular nature of the gene, and that explained, how it functions and everything, and just addressed a lot of questions. And since we reported that, of course, everybody's piled on, and now there's been dozens of reports of gametophytic incompatibility systems. They're really well understood. If you're interested in that, there's a lot of information about it now, and you should go look at it. So, okay, I just calling that kind of the end point. That's where I'm leaving off. And I'll just reiterate the take-home messages for you. Mentors and role models are helpful. Focus on the problem, not on the methods. Soft skills like leadership management styles are learned, so I encourage you to take courses and read about those things if that's important to you. Know your customers. You have to understand who benefits from your work. And also be alert, be aware of current events, and try to keep your research relevant to them. So I hope you find those messages important. This is a picture of our most recent picture of our lab group. These are great people. I was so privileged to work with these people. It was wonderful. Yeah, and I want to say thank you to a lot of different people. I couldn't mention everybody in my talk, but I'm so grateful to my mentors, role models, teachers, students, supervisors, supervisees, colleagues, cooperators, co-authors, funding agencies, stakeholders, administrative support professionals, family and friends, and to you for all your attention. And so here I am now at the end of my career. I have one more thing to offer you, and that is my shoulders, so that you can stand on them and see farther than anybody has seen before. Thank you very much. I'd be happy to take questions if anybody wants to, if we have time. I'm not watching the clock. I think we have a few minutes. Michelle.
Very nice talk, Paul.
Thank you.
So you mentioned, being aware of current events. So science policy has changed a lot in the last, say, 7 months. What advice would you give to students who are looking at all these challenges and all of these changes in scientific policy?
Yes, that's a great question. Michelle asked, what advice do I have to students who may be impacted by current scientific policies, right? I guess my best advice is stay focused on the problem. The important problems are not going to go away without research, and they're going to stay important. And if you stay focused on the problem, be creative, find ways to do the work that you want to do, and address the problem. That's the best advice I can give. Thanks for the question. Anybody else? Shenming.
And sometimes we heard that ARS is mission-driven research.
That's right.
And sometimes, I can see that some of the faculty, scientists also actively try to seek funding. But in your case, that ARS, stopped you from pursuing the other earlier research. How do you manage that transition? I guess if your mission is more broadly defined, not defined, sharing experience on that.
Right. That was okay. So Shenming asked about how you stayed mission focused when, as what happened in my case, the mission kind of changed on me. And how do you handle that transition if the mission changes? And you know, if you work in a company, that kind of thing happens every couple of years, right? So that's really a really relevant question, especially if you're employed in industry, because their mission focus drifts around all over the place. Yeah, it's a difficult problem, but you have to stay on mission, whatever the mission is. You have to be creative. Maybe, you know, reframe your problem and think about how it impacts the mission. I don't have a great answer for that. When the mission changes, you have to adapt, and it can be difficult. So maybe prepare yourselves for that, especially if you're going into industry.
I think also be aware of what you have to offer.
Oh, that's a good point.
Is like, it's what you did, is you figured out what you had to sell that other people might want, that you could sell, and then do the things you wanted at the same time.
Yeah, could everybody hear? Michelle says, be aware of what you have to offer, and I missed.
What you have to offer.
Yeah, leverage what you have to offer to address the current mission. Make sure that you have something to bring to the table. You got to figure out what it is and apply it to the mission. Is that good? Okay. Yeah. Anybody else? Yes.
And the question is, which part are you most passionate about the plant breeding?
Which part of plant breeding am I most passionate about? You know, I've always been interested in nutritional quality, and there's a little bit of a conflict there because nutritional quality isn't the most important trait in the U.S., right? It's really an international, global problem, and solving global problems is good for the U.S., right? And so I still think it's an important thing for U.S. researchers to do because you know, we're citizens of the planet. So I have to say nutritional quality is where my heart lies. Although, I mean, there are a lot of interesting problems out there, and yeah, there's no shortage of interesting things to work on. Anything else? Jianming?
When you show that lesson number 2, leadership and personnel management are learned skills, that, you know, to some people who do not have enough interaction with you, they might not like have any feelings about that, right? When Kendall was here, he shared some of the last details about how he worked with you to solve some of the student issues. You know, I had experience with working with you, which I took the easy way out, right? And you stayed to finish the students. So in retrospect, I regret I didn't go the high standard, high road. I invested my time, but you did an excellent work.
Oh, thank you.
Finish that student. I want to publicly acknowledge that. But can you share more like details? You know, because when we had a reception, Maria was giving a short speech about the number of students, number of many things you did. Right? Can you share a little bit of details so that we can get connected with that lesson number 2?
Okay. Yeah. So this is a little difficult. So Jianming is asking about leadership being a learned skill, and I was a little worried. I thought he was going to say, "Can you give me a list of books that you've read on the subject of leadership?" And actually, I know it's a learnable skill, but I didn't say that I had learned it, right? I know it's possible to learn it. Yeah. So I think so. The question was about, you know, how do I like to manage students in my group? And I mean, it's, I don't know that, I guess I've developed a style that works for me, and it's, and I don't know if it would work for anybody else, but I mean, I always think of myself as a little bit of a helicopter parent with my students, and I spend a lot of time with them. I try not to spoon feed them and answer their questions, but I do think it's important to observe what they're doing and talk about what they're seeing, because it takes a while to develop an eye for data and for research results. And sometimes, often, students do a great job on experiments and they don't get the results that they want. And they just need a little help understanding that there's not a problem with the way you did things. It's just that there's a problem with how you're interpreting your data. And I think helping my students understand their data has been one thing that helped me get students, to help students have success. And I don't know if I'm answering your question, but that's something that I always felt was important, was spending time talking about what you're seeing in your data, because there are always surprises in data. And it takes practice to figure out what your data is telling you. Yes?
I was when did you get your first funding?
My first funding?
Yes.
Yeah, so when I was a graduate student, I wrote a little proposal that got funded and helped to fund me at graduate school. And I also got a fellowship to go to my postdoc in Denmark. And then I started here in 1996, and in 1998, I got a cooperative agreement in place with Pioneer. And since 1998 through 2005, I've had external funding that whole time from different sources. And it's made a huge impact on what I could accomplish with in my work. Yeah, ARS is great because they give you money to do research, and you can keep your job without external funding, but it makes it a lot better if you can get external funding. And that's something that has really been important, and I really benefited from all my cooperators at Iowa State because I'm surrounded by all these great scientists with interests similar to mine, and we were able to put together, I don't know how many different Iowa State faculty members I've cooperated with, but it was a lot. And that's what part of really what made my career fun and interesting. Yeah. Yeah.
Thanks. Like you witnessed several students graduating while you were here, and you have seen a lot of oral presentations like defense, and then their final other presentations and seminars. So I'm curious, like as an Iowa State graduate student, what you have seen like a common, maybe a lacking skill, or like, okay, you might have felt like my students might have been developed this skill would have been helpful for them, or something, a commonality among our department students which we need to improve.
Yeah, that's a difficult question. So yeah, I have sat on a lot of committees, I think more than 150, and you'd think you'd learn something after reading 150 PhD dissertations, but yeah, it's a lot. It's a big body of knowledge. And you asked about what is different from in Iowa State about other places, maybe. I mean, Iowa State is a strong research institution. Our students are amazing. Our faculty is amazing. It's just when you're talking about agriculture, yeah, this is a great place to come. I think you all made good decisions in coming here if you're interested in agriculture. I don't know, I can't say that there's a weakness. You know, individuals have strengths and weaknesses, but as a whole, it's just been amazing to see the work that has happened here at Iowa State University over the last 30 years. Yeah, I hope you all are happy with your decision to come here, because I'm sure it's it will serve you well. Your education will serve you well. Yeah. Anything anything else?
The people online says appreciation to your work, and not for him. I think it's all the students and faculties here. So Dr. Siddiq says, great talk, Dr. Paul. It's always been a pleasure to have interacted with you. So thank you so much.
Thank you, Sadiq. That's very kind.
Paul, I think you should disclose some more numbers, because I didn't take my notepad and pen during the reception. Maria disclosed 150 patients, more than 150, right? What are the other two numbers, or one or two numbers you had?
Numbers?
Number of students you trained in your group.
Oh, I don't keep track of how many students I trained. I'm not sure.
She said that number.
Yeah.
Large number.
I don't think it's not nearly as many. Like I'm sure you have trained more students than I have. I go for quality, right? Not quantity.
150 kids is not a huge number.
Yeah, yeah. Okay, well, thanks very much for attending. It's really been fun to talk to you today, fun to work with you. Good luck in your careers. Thanks very much.
Thank you everyone for coming. Have a great rest of your evening.
None of this stuff is mine. So.
Great presentation. Thank you.
Thank you.